I'm Jon Wolpaw. I'm the president of the new society, the BCI Society, that's sponsoring this meeting. I too want to welcome you to the 6th International Meeting. Now, the first international meeting was in 1999. There were 40 people there. We have something over 400 registered for today, so clearly it's been successful. The first meeting started with the impetus of Louis Quatrano from the NIH. He was a program officer in the National Center for Medical Rehabilitation Research, and he contacted us at Wadsworth about developing a workshop on this now very beginning field of brain-computer interface research. And the emphasis was on making it a workshop. So there were basically 3 features. One, that it would take place in a fairly isolated location, so everyone would be together pretty much full time for the meeting, so there would be a lot of interaction. Second, that there be as many young people as possible, so there was money that was allocated for students and postdocs. And the third was that the program be very interactive, so that we developed the workshop format that was sort of generated really from the population, from the participants, and stressed interaction. And as the meeting's grown, we've pretty much, I think, been able to maintain that format, and those 3 main features. We're at an isolated location. We have many young people and support for them coming, and we have still the interactive workshop format as a big part of the meeting. Now, the meeting, as it developed, really owed a great deal to 2 people. For the beginnings, the first 4 meetings, that person is Teresa Vaughn, who I know is here somewhere. She would stand up. She's at the back. And for the most recent meetings, as I think you know, Jane Huggins. They've really carried the development of the meeting over the past now 17 years. This meeting is also the first one that's under the auspices of the new Brain-Computer Interface Society, which was conceived here at the last meeting in 2013. These are the board members of the society, and you'll see them walking about. Most of them are here. The person most responsible for the society is Nick Ramsey, who's now the president-elect, currently the vice president. He was the one who really organized it, took care of all the formal logistics that was essentially to establish this. He's the one who's carried it forward, and he's responsible really largely for the fact that there's a society now. So Nick should be acknowledged as well. So as I said, the meeting was conceived here 3 years ago. It was really born at Lincoln's Waffle House in Washington, D.C. about a year and a half ago at the neuroscience meeting. The board had their first meeting at the counter there. It was a very historic location. Lincoln's Waffle House is located across the street from Ford's Theater, where President Lincoln was shot, and it's a few doors down from the house where he died the next morning. So it is a historic location, and we can only hope that in a few years there might be a plaque there marking the foundation of the BCI Society there to make it even more historic. The other thing, of course, that's appropriate is that BCI researchers do a lot of waffling around. So I'll leave that on for a while. So what we want to do tonight, and fairly briefly and very hopefully effectively, is to review where the field came from, how it developed, why it developed now, where it is now, what some of its major issues are, the really important things that have to be considered as this burgeoning field moves forward. So people know, I'm sure everyone here knows, that the brain-computer interface research and development is a very new field. It only goes back about 30 years or so. It's clearly in the area of neuroscience. What people, I think, know less well is the neuroscience itself is really a very new discipline. It only goes back about 200 years, and that was when people finally figured out what the brain did. Before that, they thought things like the brain produces mucus or the brain is responsible for cooling. It was a very mysterious organ. Just as an example of how mysterious, really a little over 200 years ago, people thought the cortex didn't do anything. It was referred to as the rind, like the skin of an orange, the part that you throw away. It was thought that it really had very, very little useful function. That's, as people here know, that's changed a great deal. In fact, there are many people now who tend to think that the cortex does everything, which also is not entirely true, but it's clearly an improvement over 200 years ago. But basically, at that point, people figured out that what the brain does is it interacts with the environment. It interacts with the environment to serve the needs of the person or of the animal. And it became clear pretty rapidly that electrical activity was important in this, that the brain produced electrical activity, and therefore if you could look at that activity, that you could learn something about the brain. And in the course of the 19th and the first half of the 20th century, two major streams of ways of doing that developed. The one developed in the 1920s was led by Hans Berger, who was an early neurologist and psychiatrist, and he showed in 1924 that it was possible to record brain activity from the scalp, EEG activity. It's interesting that he first did this in 1924, but he didn't publish anything till 1929. He worked on it for 5 years before he published anything, and then he published the first of about a dozen papers that established the field of EEG research and clinical use. So this is a picture of Berger and fairly blurry initial traces. I left this, I know this is a low resolution photo, and I was going to get a higher resolution one, but then I was reading something about Berger. Up till recently it was believed that he had really lost his laboratory and been forced to retire by the Nazi government in Germany in the late 1930s. More recent research has shown that he was a little bit more of a sympathizer with the Nazis than we might find comfortable, that he was not really as opposed as many other German scientists were. So for that reason, I've left this a little blurry. But he did very clearly do something very important scientifically. So the other major stream developed in the first half of the 20th century, which was recording individual neurons. Neurons became identified and were identified as basic structures in the nervous system by Cajal and Golgi in the late 19th century, and there became a great focus on recording their activity. And this became possible in the middle of the 20th century, first in anesthetized animals, then in awake behaving animals. One of the early people doing that was a man named Birdsey Renshaw, that many people may know his name. The Renshaw cell in the spinal cord was named after him. He is unfortunately also distinguished by the fact that he died very young. He died at the age of 37 in 1948 from polio, only a few years before the polio vaccine became available. So these two streams developed, and they were basically used both for research purposes, EEG and single neuron recording, as well as in the case of EEG for clinical and purposes. At the same time, there was some mention made, sometimes kind of jokingly or fancifully, sometimes seriously, that these same signals might be used for another purpose, that they might be used as brain outputs, so that instead of muscles, it might be possible to use these brain signals to do things, to enable people to act through electrical activity. And that's shown here in a holiday card that Hans Berger received in 1938. It was sent by Herbert Jasper, who was that time very young. He became a very distinguished North American clinical neurophysiologist over the next 60 years. But he sent him a card showing a picture of himself with some EEG electrodes and EEG activity, and the EEG activity is saying, wishing you a pleasant Yuletide, a new year as you like it. So here was sort of a fanciful rendition of the idea that EEG could be used as a means of communication. And that developed more in the last 20, 30, 40 years in both of the major streams, both single neuron recording and EEG. And we have here two examples of this basically living history and still very active scientists to tell us about that those initial events when the first BCI efforts got underway. The first presentation in regard to single neurons will be by Eb Fetz, who has been at the University of Washington, the National Primate Center now for about 50 years, has done very distinguished work looking at neuronal activity primarily in the cortex in many different ways from basic science perspectives, as well as for the possibilities for using it for BCIs and other purposes. He's really one of the longest lasting really and most distinguished people in the field of single neuron recording. So Eb will tell us something about what happened in those early days.